Alright, this is a continuation of the previous recording on the urinary system because the file apparently got corrupted and cut off. So this is just a continuation from that. So this is a more highly magnified view of the cortex in the kidneys, and you can tell it's cortex still because you can see numerous renal corpuscles here. But to really look at the important structures with the renal corpuscles, we need to use a higher magnification. So here's a higher magnification view of several renal corpuscles, and at this magnification you can see details such as the glomerulus here, which is surrounded by two layers of connective tissue capsule. The outermost layer of the capsule is called the parietal layer, and it's made of a simple squamous epithelium, as you learned early on. The innermost layer, called the visceral layer, which makes direct contact with the endothelium of the glomerulus, that's called the visceral layer, and it's composed of specialized cells called podocytes. You can read more details of the morphology and function of podocytes in your basic histology text. We also have a model in lab that shows their structures. So Bowman's capsule, again, you have visceral layer composed of podocytes directly making contact with the glomerulus, which is a capillary tuft, and then you have the outer layer called the parietal layer. So both those layers are Bowman's capsule. The glomerulus, along with Bowman's capsule, collectively is called the renal corpuscle. So there's often confusion by students about what's the difference between the glomerulus and the renal corpuscle. The glomerulus is just the ball or tuft of capillary. The renal corpuscle includes that as well as Bowman's capsule. You can also see various tubular structures in mostly in cross section, but some in more longitudinal section. There are a variety of different tubular structures in the medulla and cortex that you need to be able to differentiate based on their structure. So before we do that, let's take a higher magnification view of the renal corpuscle and talk about some of those structures. You can see surrounding the glomerulus there is a region that appears to be white. This is basically empty space. This is known as the urinary space, and this is what fills up with urine as the plasma filtrate accumulates as it passes through the filtration slits across the glomerulus. It pulls into this space. So here's where urine initially forms. It then will pass into a network of tubular structures. The first tubular structure it passes through is this structure here, which is called the proximal convoluted tubule. The proximal convoluted tubule gets its name because of its convoluted nature and because it is at the proximal portion of the nephron. So right here you can actually see the proximal convoluted tubule connected to the urinary space. You can see the lumen of the PCT is continuous with the lumen of the urinary space right here. Not every section of a renal corpuscle are you going to see the lumens connected to each other, but sometimes you can. This region where the PCT attaches to the renal corpuscle is called the urinary pole of the renal corpuscle. It's called a pole because it's at one side. As you might suspect, there's another pole at roughly the opposite side, and this pole is called the vascular pole. The vascular pole is where you have the afferent and efferent arterioles that join with the renal corpuscle right here. The afferent arteriole brings blood in towards the glomerulus. As the blood passes through the glomerulus, forming some urine filtrate, then it leaves through the efferent arteriole. You do not need to worry about distinguishing between afferent and efferent arteriole on these sections, but you do need to identify the vascular pole, which is where these arterioles actually attach to the renal corpuscle. So this is a really nice section because you can see both the urinary pole and the vascular pole attached to the same renal corpuscle. Many times you may see only one or the other, or you may see neither. So it just really depends. Another important structure you need to know is the distal convoluted tubule, and you see a distal convoluted tubule here on the opposite side, in this case adjacent to the vascular pole of the renal corpuscle. Notice that, like the proximal convoluted tubule, the distal convoluted tubule is also made of a simple cuboidal epithelium, but notice that the distal convoluted tubule epithelium is a lot flatter. So these are smaller cells, and there's less cytoplasm, so they're flatter, and the nuclei are spaced closer together. In the proximal convoluted tubule, the cells are a lot bigger, the nuclei are spaced farther apart, and the cells are taller. So they're both simple cuboidal epithelium, but there are some differences in the shapes. That's one difference. Another way to tell them apart is that the proximal convoluted tubule often has a kind of a dirty appearance in these types of sections, whereas the lumen of the distal convoluted tubule appears a little bit cleaner. A third difference is that the proximal convoluted tubule actually has a brush border epithelium, so there are microvilli on the apical surface of these cells, but there are no microvilli on the apical surface of the distal convoluted tubule. Unless you have a really good section at high magnification, you may or may not be able to distinguish the epithelium on the apical surface between those structures. So, but you can use all three of those as potential landmarks to distinguish between those structures. In this case, it's also really easy because you can see where the PCT directly attaches to the renal corpuscle, but oftentimes you don't actually see those structures attached to the renal corpuscle. Notice on the portion of the distal convoluted tubule that's closest to the renal corpuscle, which is this portion right here, the wall, it looks a little bit different than the wall over here. More specifically, these cells are more columnar in appearance, so the epithelium appears much thicker. For this reason, this structure is called the macula densa. So the macula densa is a portion of the epithelium of the distal convoluted tubule that is taller and makes a close association with the afferent arteriole that feeds into the glomerulus here. The macula densa and the portion of the afferent arteriole that it makes contact with actually are important structures because they regulate blood pressure by regulating secretion of angiotensin and renin in the renin-angiotensin system. So the cells in the macula densa possess chemoreceptors that sense salt concentrations, which it can use to indirectly assess the blood pressure of the body and regulate renin secretion, which is a hormone that's secreted by cells that are found in the afferent arteriole. The afferent arteriole also possesses stretch receptors, so it has a direct way to measure blood pressure. And together, certain cells called granular cells within the afferent arteriole and the macula densa cells of the distal convoluted tubule interact with each other to regulate renin secretion, which ends up regulating blood pressure levels. Because these two tissues interact with each other, the macula densa and afferent arteriole collectively are often referred to as the juxtaglomerular apparatus. That's a mouthful, but if you think about what it means, it actually makes sense. Juxtaglomerular means that it's next to the glomerulus, so that makes sense. The cells that actually secrete the renin are called granular cells or juxtaglomerular cells. This is also renal cortex, but you don't see a renal corpuscle in view here, but instead you see different sections of proximal and distal convoluted tubules. Now, because they are not directly attached, you don't see them directly attached to the renal corpuscle here, you have to rely on other characteristics to differentiate between them. But again, the PCT has a much thicker cell wall than the DCT. So this is a proximal convoluted tubule. Here, 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 here. Most of these tubules are proximal convoluted tubule, but here you see two sections of distal convoluted tubule. Again, you can look at other landmarks as well, such as the fact that the lumens of the PCT, again, tend to have a dirtier appearance. This is a cross-section through the medulla of the kidney, and you can see it has a different appearance, although there are still many tubular structures that are sectioned through here. So there are several tubular structures you need to be able to identify and differentiate between. And what complicates this is that at least two of the structures are again composed of simple cuboidal epithelium, but there are significant differences between them that make it easy to identify. This structure right here is a collecting duct or collecting tubule. Either term is acceptable. And the collecting duct can be identified by the fact that it's a tall cuboidal epithelium. It's still classified as simple cuboidal epithelium, but you can see it's a bit taller, and so it has some characteristics in common with columnar epithelium, such as the fact that the nuclei are located a little bit more closely to the basal surface. Another distinctive feature of the epithelium of collecting tubule walls is that the cytoplasm is very pale, and that allows you to see the lateral borders between the cells quite clearly. So you can see these lines between the cells as the lateral borders. These are the only structures in the medulla of the kidney where you can see the lateral borders. So here we can identify this structure as a collecting duct or collecting tubule. Here's another collecting tubule. Here's another one over here. And here's another one over here. And you can see part of one sticking out here as well. The other major structure that has an epithelium composed of simple cuboidal epithelium are the thick portions of the loops of Henle, which are many of these structures. Here you see one here, and here, and here. How do you distinguish them from collecting tubules? Well, there's a couple landmarks. Again, the cytoplasm tends to be more grainy. It is less pale, so it's darker and more eosinophilic in comparison to the cytoplasm of collecting tubules. Also, because the cytoplasm is less pale, you really can't make out the lateral borders between cells like you can in the collecting duct. Another difference is, again, that the epithelium is flatter, so it's a flatter cuboidal epithelium. The cells are less tall, and the nuclei are located more in the center of the cytoplasm rather than closer to the basal surface. So those are several landmarks you can use to distinguish between the thick portions of the loop of Henle and the collecting tubules, both of which are tubular structures in the medulla that are composed of simple cuboidal epithelium. There are also thin loops of Henle in the medulla of the kidney, and they get their name because the wall is thinner. And in fact, the walls of the thin loops of Henle are composed of a simple squamous epithelium. So this is a thin loop of Henle for an example. You can see the cells are very flat. You can see some of the cytoplasm. Here's the nucleus, but notice the nucleus is a little bit squished up here. So these cells are flat and squamous in morphology, but they have relatively large lumens. These are not the only tubular structures in the medulla, though, that are composed of simple squamous epithelium, because you also have lots of capillaries because the kidney is highly vascularized. This is a capillary to the left of this loop of Henle structure. It's also composed of simple squamous epithelium, but it's even flatter. So you can actually see some of the cytoplasm here in the thin loop of Henle cell, but in the endothelium it's hard to see any cytoplasm. Also, the capillaries are a lot smaller in diameter and have much smaller lumens. So make sure you can distinguish between thin loop of Henle and capillaries in cross-section. Here's a capillary, here's another one, here's another one, here's another one. This is a thin loop of Henle. Here we have thin loop of Henle down here and over here. So take some time to practice in lab being able to distinguish between those various structures. Those are the major structures you need to know in the renal medulla. Here's another section of renal medulla. You can see three collecting ducts or collecting tubules. Here's one here. Here's another one. Here's another one. You can see multiple thick portions of loop of Henle here, 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 and so forth. You can see multiple thin portions here, here, here. You can see capillaries as well. Be able to know at a glance if you are looking at a particular part of the kidney, whether it's cortex or medulla. So for an example for this slide, we could ask, is this in the cortex or the medulla of the kidney? This is pretty easy. It's the cortex. You can tell because you can see the renal corpuscle. So in general, if you see renal corpuscles and proximal and distal convoluted tubules, you're looking at the cortex. If you're seeing lots of collecting tubules and thin and thick loops of Henle, you're in the medulla. This is a section of the medulla showing the collecting tubules, what they look like in longitudinal section. And again, in longitudinal section, you really get a good idea of how tall these cuboidal cells are. They're almost columnar in morphology. And again, you can easily see the lateral borders between the cells, making it really easy to distinguish between cell types. Here's a section where you have a collecting tubule in longitudinal section. Here's thick loop of Henle in longitudinal section, and here's thin loop of Henle in longitudinal section. So be able to identify those structures both in cross section and in longitudinal section. So that's what you need to know for the kidney. This is a cross section of the ureter, which is the thick muscular walled tube that brings urine produced from the kidneys and drains it and brings it to the bladder, where it's stored until urination. The ureter, we're not going to go over detailed histology of the wall of the ureter as we have with some tissues. Basically, just be able to identify it. You can identify it based on its highly fold mucosa that contains a transitional epithelium. And the lumen is fairly narrow. It has a very thick muscular wall composed of lots of muscle, and then you have an adventitia that surrounds that. So that's all you really need to know for the ureters. This is the urinary bladder. We've looked at this structure early on in the lab because it also possesses transitional epithelium in the mucosa. So the epithelium is just the small superficial layer of the wall of the bladder that you can see here. If you look at it more closely on the microscope, you can just identify the transitional epithelium. Most of the wall of the bladder is actually muscular, or composed of connective tissue. So underneath the epithelium, which is this portion here, this is all composed of muscle, and deep to that you have connective tissue. So that's the wall of the bladder. Be able to identify that. And then finally, the distal portion of the urinary tract is called the urethra. And so the urethra has a different appearance and different distribution of epithelium in the male or the female. So for the urethra, you need to be able to identify it as the urethra and also identify whether it comes from a male or a female. This is a cross-section through the penis, and so this is a male urethra, obviously. It has a fairly narrow opening, and the epithelium of the male urethra is mostly lined with PCE, that is pseudostratified columnar epithelium. It's not ciliated, so it's not PCCE, but it's PCE. The distalmost portion of the urethra, however, is composed of stratified squamous non-keratinized epithelium. But most of the epithelium in males is PCE. You really need to look at it at a higher magnification to distinguish between them. This is the female urethra, and it has a much wider lumen. It's also much shorter, And the female urethra also, parts of it are composed of PCE and parts of it are composed of stratified squamous non-keratinized epithelium. However, the relative proportions are basically inverted relative to the male urethra. In other words, most of the female urethra is composed of stratified squamous non-keratinized epithelium, which you actually can see pretty clearly at this magnification. Only the more proximal portions are composed of PCE. So that's it for the urinary system, and that's what you need to know.